Hi, I'm Tom Lynch, and welcome to Science Says. Whether this is the first one you've been to or you've been to many of them, I want to welcome you. This is a wonderful chance for you to learn about the incredible science that's happening here at the Fred Hutch. And I really look forward to spending the next hour together and answering questions. And we look at this as very much an interactive opportunity for you to ask questions and for us to try to address some of them. So as everybody knows, or maybe doesn't, there are 6 big themes to science at the Hutch, and today we're going to explore one of them in great detail. Just to refresh your memory, the 6 big themes are looking at the immune system and exploiting the immune system to understand and treat cancer. This has proven to be incredibly important in leukemia, lymphoma, cellular therapies, bone marrow transplant, all great examples of that. The second is precision oncology, understanding what genes drive cancers. Important in breast cancer, important in prostate cancer, important in lung cancer. Third is the idea of prevention and early detection. We have one of the most incredible public health divisions here at the Fred Hutch that's looking at what are the behavioral factors and the lifestyle choices we make that influence our ability to get cancer. Really, really important. Fourth is the idea of viruses. You're going to hear today about some of our work in COVID and our ability at the Hutch to work on COVID after having done so much in the area of HIV is something I'm extremely proud of in our scientists. And next is the commitment. Fifth is the commitment that we make to diversity, equity, and inclusion in our science, in understanding why outcomes are different for different populations and trying to understand how we can make science and our cures more accessible to more people across Seattle, across Puget Sound, across Washington, across our country and our world. Today, we're going to focus on the 6th pillar, the intersection of tech, data, cancer, and virology. And it's really something which I'm incredibly proud of. We're here in Seattle, which is the epicenter of where technology and biology meet and interact, an incredibly rich place to be doing this. So the title of today's talk is Tech, Terabytes, and Transformation, supercharging cancer research with new tools and approaches. And so you're going to have a chance to talk to some of the scientists who are on the front line of using some of the most innovative new technologies and tools to understand cancer and to move forward. If you think about it, vaccine makers were able to move quickly because of the work we have been doing in understanding how genes cause proteins to be made. Really remarkable work. And the same forces that we saw that pioneered and empowered the advances in cancer, in viral vaccines, are also applicable to cancer biology. We're going to talk about some of that today. So what are some of these new technologies? Next generation sequencing, our ability to mine massive amounts of data. You're going to hear from some of our data scientists. We're talking about enormous amounts of data that we could never have imagined in the past that we'd be able to manipulate, understand, and visualize. So this ability to look at data is making a huge difference. It's transforming how we look at molecules, how we look at organs, how we look at the body in general, how we can figure out what's really happening with a disease, how we can possibly diagnose disease earlier. And you're going to hear from scientists who are working on early detection as an important part of this. So I'll be joined shortly by 3 Hutch researchers who are doing exciting work: Gavin Ha, Melody Campbell, and Rob Bradley. Later in the program, during our COVID corner, I'll be joined by one of our COVID experts, Dr. Jim Kubin, and Jim will give a brief update on where we are with COVID vaccines and will be available to answer questions. So I'd like to bring the first scientist up, Dr. Gavin Ha. Gavin is one of our newer researchers. He's part of a large cohort of faculty that we recruited in the last couple of years that are accelerating our work at that intersection between computational biology and cancer biology. He's a computational biologist himself. He's also a geneticist. He studies the role of genomic alterations in cancer. He and his team are developing tools to detect minuscule amounts of tumor DNA that's circulating in the blood. And that's also the expression that's been used to describe this are liquid biopsies. If you think about it, this has the potential to transform not only cancer treatment and how we follow cancer patients, but also how we diagnose cancer and how we screen for cancer. It may be that we will have blood tests that will replace mammograms and colonoscopies and PSA tests and the kind of things that we're doing now to try to diagnose cancer earlier. Maybe we'll have blood tests that will do this. So Gavin, welcome. It's really great to have you. And so, Gavin, liquid biopsies are something we've wanted to do forever. The idea that you could just get a blood test that could help diagnose cancer and tell us that someone has a cancer in their body could be incredibly exciting. Tell us about the potential and what you're doing in this area.
Right. So thank you, Tom, for the nice introduction and an opportunity to tell you more about our work. So we're excited about the potential applications of liquid biopsies, such as from the blood, as you mentioned, especially when we think about developing diagnostics for cancer that are both, non-invasive, easily accessible, and already routinely collected in the clinic. So being able to detect the presence of DNA released from tumor cells, which we also call cell-free DNA because it's outside of the cells now, provides a window into the entire course of disease for a patient. It can start from, for example, applications in early diagnosis, either detecting the tumor at an earlier stage, or perhaps even before symptoms arise, which I think maybe you termed, a holy grail of sort of cancer detection. But there are also other critically important applications, such as monitoring for tumor recurrence. That is, seeing whether the cancer has returned, say, after surgical resection of the tumor, as well as monitoring patients' response to therapies. But we can do more than just detect and monitor cancer. We can also identify mutations and genetic alterations in the DNA, which can serve as biomarkers for predicting how a patient might eventually respond to certain therapies. And we hope that this can help inform treatment decisions.
And so tell me what's involved. And remember that we have a really diverse audience. We have some people on this call who help to design sequencing machines and understand sequencing probably better than you or I could ever. And then there's other people who are lay people and don't do genomic sequencing for a living. So how does a liquid biopsy work?
Right. So obviously it starts with a blood draw from an individual who has cancer. And of course, in the case where you want early detection, perhaps before cancer arises. But let's say for now, the example of patients with cancer, blood draw happens. What we do is we actually isolate the plasma component. So this is blood plasma. So we basically get rid of all the blood cells, red blood cells, white blood cells, and we take what's free floating in the plasma. Okay, and then we take that and we sequence it, as you mentioned. So really, a lot of this work requires performing genome sequencing, and then that generates large amounts of data for each of the different fragments that we actually obtain from the blood.
And how much of this is work that could be done anywhere versus how much of it is work that requires real sophisticated ability to analyze the data that comes out of the sequencer?
Right. And, so part of my work and our excitement, in this area is that, we want to make this broadly accessible and widespread, easily used by, communities, not necessarily at a specialized institution or hospital, but be able to use as a test for diagnostic in areas where you don't have to have such sophisticated machinery. And the way that we are approaching this is really through sort of, as you mentioned, through data science and analysis, right? Computational approaches. The processing of the sample is becoming quite routine. Now it's just how can we extract the information that we want from these samples.
And so I see, Gavin, today that you're seated in the steam plant, and I can look out and we can see Lake Washington right behind you. So it's great for those of you who have helped support the establishment of the steam plant at the Hutch. We are really excited by this new facility. And in the steam plant, computational biologists, Gavin, like you, are right next to people who are actually doing sequencing. And tell me a little bit of how that's working at the steam plant.
Right. So, it's been obviously during the pandemic, it's been for us, we're a computational focused lab, and so we haven't had the opportunity to come in and interact as much, but we're hoping that as things sort of relax, that we can do that. I think being integrated in this kind of environment really accelerates the science even more because of the collaborative nature. And different computational tools that we use, machine learning, deep learning techniques, require large amounts of data to be generated, but that also has to be generated in such a way that fits in with the modeling that we do. So it's really an iterative process that we go back and forth with people who are generating the data and people who are analyzing the data.
Yeah, and obviously, I think your point is well taken. That once we're through COVID, the ability for us to be able to interact personally is going to make a big difference. And obviously, that's something we can't do right now, but very much look forward to those kinds of connections that we make. Last question that came in actually from one of the people listening is, what about false positives in the blood? There are some studies that have shown that many people have evidence of some cancer markers in the blood that don't necessarily mean that they might have cancer. There may be clonal abnormalities in some B cells. There might be some transcripts of RAS that might be mutated that might still be in the blood in people who never develop cancer. What do you think about that possibility as we develop liquid biopsy technology?
Yeah, that's a very good point. I mean, I think it's both, specificity, but also sensitivity, sort of what we care a lot about, because we're trying to find small amounts of molecules. But of course, the specificity matters too. You don't want to have false positive predictions here. And so, one of the things that you alluded to is this observing mutations in blood cells in healthy individuals, right? And so that kind of contributes to what we see in liquid biopsies. And so there has been a lot of effort to try to reduce this. So obviously, it's some active area research that we're trying to be very careful about what we say. So we want to be able to have very, very high specificity. And that's kind of what we try to optimize. And then score sensitivity gets at as well. And so, again, this is sort of, a non-invasive test. So there's obviously a lot of advantages to this. It's not necessarily routinely used in the clinic now, but we're working on ways to try to improve the performance so that it is then suitable for clinical diagnostics.
Great. So, Gavin, thank you so much. We're going to have you back for questions in just a few seconds. While we're waiting for Melody Campbell to come up, I'd like to take this opportunity to thank Laird Norton Wealth Management for your support. And we want to thank them for their support of science at the Fred Hutch. It means a huge amount to us. And thank you very much for enabling the science that you're hearing about today. So next, I want to bring Melody Campbell up. Melody, welcome to Science Says. The great part of Melody is that, Melody, you were the first recruit that I had the chance to talk to when I started a year ago. And I remember one of the first phone calls I made, Melody, was to you, welcoming you to the Fred Hutch. So I am incredibly happy that you're here with us. And again, we have a very diverse group with us today. Melody comes from the Basic Science Division, and I want to also give a shout out that this is the 40th year of the Basic Science Division at the Fred Hutch, which is a remarkable accomplishment. Some of our most incredible scientists are in the Basic Science Division. Melody joined us in July, and she's the inaugural director of our cryogenic EM facility. The CryoEM facility is called for short. This is a groundbreaking microscopy technique that allows us to look at proteins and develop 3D models of proteins at the atomic level. So if you could imagine, a patient comes in with cancer and we're able to figure out what genes are abnormal in that cancer. People like Melody are able to figure out, well, if the gene is abnormal, this is what the protein might look like. And so they can design both theoretical designs, and they can also look at the actual proteins and figure out what they look like at a molecular level, which to me is absolutely extraordinary. And her own research focuses on how cells communicate and interact with their surroundings by understanding their geography and their morphology. So Melody, again, tell us a little bit about cryo-EM in a way that we can sort of understand it. As I said to Gavin just a few seconds ago, some people on this call probably are experts in cryo-EM, and some have never heard about it. So tell us a little bit about it.
Okay, thank you so much for the introduction. I remember that welcoming phone call very well. So as you mentioned, cryo-EM stands for cryogenic electron microscopy. And it's a set of techniques that we use to look at things that are too small to see in very high details with a more conventional light microscope, such as viruses, DNA complexes, or individual proteins. And the way it works is that instead of shining a beam of light at these samples, we shine a beam of really high energy electrons. And then this is where the cryo part comes in. So the electron beam is actually really damaging to biological samples like proteins or viruses. So we have to freeze these samples at a really cold temperature, almost negative 200 degrees Celsius, which is over negative 300 degrees Fahrenheit. And what this does is we trap these proteins then in a really thin film of a special type of non-crystalline ice. So this non-crystalline ice not only protects the protein such that we can image it, but we don't have to add any type of special chemicals or stains. So we've literally frozen this protein in something which is kind of like a native environment. And then I guess this leads to the question of, well, why do you want to look at a protein in this native environment anyway? And as Tom already mentioned, we want to look at these proteins so that we can understand how they work, because when we know how they look, that gives us a lot of clues about how they look. I mean, about how they work. And then when we know how the protein looks, we can try to fix it if something goes wrong, which is something that happens in many diseases.
And so, Melody, it was my understanding, this is a fairly complex and extremely sophisticated piece of machinery. Can you tell us a little bit about what it was like to establish a brand new center for this? Because that must have been incredibly exciting.
Yeah, it was really exciting. There was a lot of excitement here at the Hutch to get this up and running. But really, these microscopes are, they're really finicky machines. So we had to renovate the rooms, just the rooms, to put them in. Then these things are enormous. They weigh thousands of pounds. They're over 7 feet tall. So we can't fit them through normal doorways. So we needed to get these microscopes shipped in many, many boxes. And then we had an engineer on site for the past 4 months who was building these microscopes up. Additionally, each component of the microscope is very specialized. So we had to work with several different companies to get everything sorted out. For example, there are certain caveats about having a microscope like this in a big city where there's something called electromagnetic interference that will interfere with the microscope's ability to work properly. So we had a company come in and install these special cables that circle the microscope to counteract that type of interference. So it was an adventure for the past 4 months, but we've finally gotten it all up and running actually last week.
So would it have been easier for us to build a facility out near the Snoqualmie Pass where there'd be less electromagnetic interference?
I don't know about easier. That would have been quite a commute for me.
I'd say quite a commute for you. So can you give me an example of how CryoEM has revolutionized a field of biology? What's something that you can think of that some of the people watching today might make sense to them?
So CryoEM in the past decade or so has enabled us to look at certain proteins in much, much higher detail than we were ever able to look at them before. There are already other types of techniques, structural biology techniques, such as NMR or crystallography, that allow us to look at proteins in high details, but some certain proteins just don't work using those techniques. And one probably very relevant example is the coronavirus spike protein. So this is the protein that's displayed on the outside of a virus. If you're familiar with that really iconic image of the coronavirus, it's that red protein, and it's very large, and it just never crystallized. So this means that crystallography, that technique, could never be used to image it. Well, CryoEM was used to image that structure, and by finding out what the structure of the spike protein was, scientists were then able to make small changes to that protein sequence. And these small changes were then used to make that protein more stable. And by having a stabilized version of that protein, that's actually what's used now in the Moderna and the Pfizer vaccines, where based on that CryoEM structure, scientists changed the protein to make it a better type of protein that can be used in vaccines.
That's a fantastic example, and I really thank you for that. One question that came up is, you think about the world of artificial intelligence and the idea of computer modeling. Can you talk about, do you see a world where computer modeling would be used to make CryoEM even better?
Yeah, so I think that CryoEM is pretty closely linked to many computational techniques. For example, so the images we get using CryoEM are actually kind of, they're not that easy to interpret. So we have to draw on a lot of existing computational algorithms that will help us understand these images better. So I think that in that way, the two are very closely linked.
Yeah, and I think it'll be really exciting to be able to see how, with machine learning and AI-enabled techniques, how we can really even advance this field even faster. And understanding, I think that one of the things I've watched in cancer biology over my career is we now understand so many more of the proteins that are driving cancer. And now, excuse me, now it's a process of trying to understand how to interact with those and prevent them from causing cancer. And so understanding the weaknesses within the proteins is something that structural biology is giving us such a window into. And I think it's, again, something that the Hutch is just delighted to be in. It helps both our viruses, our viral work, and our cancer work, which is a great example of that. And I guess the last question for you, Melody, is how has this been taken up by some of your colleagues? You're obviously an expert in CryoEM, but I would argue CryoEM probably could be helpful even to people who aren't like you, aren't an expert in CryoEM, but care a lot about proteins.
Absolutely. Yeah. And that's part of the idea of setting up this shared resource at the Hutch, so that not only specialized cryo-EM users can do it, but so that it's open to other types of scientists, because the ability to literally see the protein that you're working with, or see the effects of certain small molecule therapeutics on the protein, or just see it in the context of whatever your biochemical experiments are really important for being able to understand it better.
Great. Melody, thank you so much. We're going to bring you back in a second to answer some questions. I'd like to next introduce Rob Bradley. Rob Bradley is a computational biologist and biophysicist. He is in our basic sciences division as well, just like Melody. He uses statistical approaches and technologies like CRISPR knockouts to understand cancer biology. In particular, he studies how malfunctions in RNA processing can lead to cancer and other genetic diseases. For his work, he was named the McElwain Family Endowed Chair in Data Science. The genetic links he discovers, hopefully, will become the targets of future therapies. Rob, great to have you with us today. Okay, so I guess the first question for our audience is, what is a computational biologist, and how is a computational biologist different from Melody, she's in your division as well, and she does cryo-EM experiments. You do computational biology. Tell me what the difference is between those two areas.
That's a great question. I think if you ask 10 computational biologists, you might get 10 slightly different answers. And the answer has changed over time. So a long time ago, it used to be that biologists who did computation tended to only do computation. That was what they did, and it was fairly specific. These days, I think it's much more organic, where a lot of biologists do a mix of wet lab experiments as well as computational biology. They might have research programs that solely rely upon work on computers. They might have research programs where they go back and forth between the bench and a computer, or they might have research programs where they both advance their own research and they also play a supportive and collaborative role to help, for example, a big organization move its research forward.
So when you go to work in the morning, okay, could you work just as effectively from a computer based at home or a small office if we built you in the Snoqualmie Pass, for example, along with the new cryo-EM facility? I mean, do you need to be in a place with other scientists? How does a computational biologist work?
It's a great question, and I'm really intrigued about the satellite facility that's going to be happening. Sounds like a great environment. I think the answer is sort of yes and no. So in theory, a lot of the computational work that I could do, that my lab can do, could happen anywhere. We could be on the Snoqualmie Pass interacting virtually with people at the Hutch working in labs and move our science forward. But I think at the same time, we're all humans and scientists, and we want to have those interactions that are possible through day-to-day interactions. Like you were mentioning the steam plant earlier, and how one of the excellent things about it is that it allows for these easy conversations to nucleate where new ideas can come forward and people can learn from each other in a really seamless way.
And so, Rob, you study RNA biology. Tell me a little bit about what you're doing with RNA and how RNA processing is important, and what are you learning?
It's a great question. It's funny thinking back to when I started learning about RNA, because nobody really thought about RNA. These days, everybody's thinking about RNA all the time. And What we study is a specific process called RNA splicing. So RNA splicing sounds really technical, and it is technical, but it's a really interesting process. So if we go back to when we learn about biology and genes in grade school, we learned that a gene encodes a protein. So genes are genetic information that we get from mom and dad. Proteins are what actually make up our bodies and make us who we are. But actually getting from a gene to a protein is really complicated. So one example is yeast that we use to bake bread. It has about 6,000 genes, and it has about 6,000 different proteins. But humans only have about 23,000 different genes. I mean, I think I'm more than 4 times as complicated as yeast that we use to bake bread, right? I think we all are. And yet somehow we only have 4 times as many genes. So what's going on? Well, one of the most important answers to that question is that in humans, one gene doesn't just make one protein. It might make 5 proteins, it might make 10, it might make way more. There's a gene in Drosophila, which is a fruit fly, that encodes over 30,000 different proteins. One gene, 30,000 proteins. So splicing is the process by which one unit of genetic information, one gene, can produce lots of different functional units of proteins.
And so how does that have anything to do with cancer? We're a cancer center. You're studying a process called RNA splicing. The people who are watching today support cancer research. How is supporting Rob Bradley going to support cancer research?
It's a great question. And to answer that question, in some sense, I think the simplest way to think about it is to think how splicing happens. So we learn about genes and genetic information like they're an abstract concept, but they're actually related to a physical thing, which is an RNA molecule. And you can think about a physical thing, the RNA molecule that tells the body how to make a protein as a message. Like if we think of it as an email, for example, an email has paragraphs. And if you take an email and you randomly insert or delete different paragraphs or swap their order, then all of a sudden you might communicate a message that's very different than what you should. A cell might be supposed to get a message that says, divide if I need more blood cells. But instead, if you delete a paragraph or a sentence, it might get a message that says, keep dividing, just keep dividing. And that's what happens in a number of cancers. So in many blood cancers, for example, there are genetic changes that occur in cells in the body, and these cause the process of splicing to go wrong, so that your message, which is supposed to say, only make more blood cells if you need them, starts saying, just always make more blood cells. So many different diseases and many cancers are actually caused by errors in RNA splicing. And one of the things that's most interesting and mysterious is there's some disorders such as myelodysplastic syndromes or other leukemias that carry the exact same mutations affecting RNA splicing that we see in a lot of other cancers, such as breast cancer or pancreatic cancer.
Great. So, Rob, last question I'm going to ask you before we move on is talking about the role of big data. So when you think about it, you started looking at data from small groups of patients, maybe 10 to 20 patients. Now you're looking at hundreds of thousands of molecules and samples collected from thousands of patients. In your career, and you're still in the early part of your career, you've seen this explosion in our ability to handle big data. What's changed and what allows us to do that?
It's a great question. It's a little bit like thinking about why we could start doing CryoEM. It required the convergence of a lot of different technologies. If we're looking at huge amounts of data, there are really a couple of things that had to happen. So the first thing was that we had to have new experimental instruments that let us generate huge amounts of data from one sample. But it turns out that then creates its own problem, because once you have so much data that you can't just load it up into a spreadsheet, the human mind can't really comprehend it. It's so much that it's overwhelming. So then we need new developments in statistics and computation in order to reduce that huge amount of data to something that we as humans can actually begin to understand. And then finally, one thing that has really happened recently is a huge number of patients have begun participating in research studies and been willing to donate samples to create these amazing repositories that let researchers such as myself and others start investigating what the genetic origins of their cancers are. So understanding what changes happened in the cell, for example, in the skin that eventually gave rise to a melanoma.
And so what's the single biggest thing? Is it the advent of the cloud? Is it what has enabled this? Because I remember just 15 years ago when we were developing a sequencing facility, and it was probably 5 miles remote from where our computational biologists were. We had to have literally a 5-mile fiber optic cable dropped into the side of the highway between the two different laboratory facilities. Tell us now about how that's working and how the cloud has enabled this.
Absolutely, the access to huge computing power is really critical, as you point out. So resources like the cloud enable us to do things like take a huge amount of data, so much that Literally in the old days, I used to have a desk drawer with 20 hard drives in it. And now instead of that ridiculous setup where I plug in hard drive after hard drive into my computer, I can just type in my laptop and then access all the data I want, which is stored on the cloud, and apply some new statistical algorithm to it just within an hour. So this easy access to big technology has made a huge difference.
That's remarkable. And again, I give credit to Bradley Smith, who also asked that question about the advent of economical cloud computing being critical to this. And Bradley, I think Rob agrees with you as well. So Rob, thank you very much. And we'll be with you in just a second. I want to bring in Jim Kublin with us. Jim is the executive director of the HIV Vaccine Trials Network. And you might say, gosh, HIV. But the thing for Jim and Larry were incredibly well positioned as physicians working in the HIV vaccine area to pivot to COVID, and they formed the CoVPN, which is a COVID-19 prevention network based at the Fred Hutch, while still continuing the work of the HIV vaccine network. We have not given up our effort and work in HIV, folks. I want to make sure that people understand that. The CoVPN has led the phase 3 COVID-19 vaccine trials as part of Operation Warp Speed. Jim is also a global health expert who works on development of vaccines and treatment for life-threatening infections like HIV, tuberculosis, malaria, and is really skilled at designing and implementing large-scale clinical trials. And we could not have been happier that Jim was the right person at the right time to be here when COVID vaccines needed to be tested. So I guess I'm going to start off, Jim, by asking you the controversial question from today's Seattle Times and New York Times, which is, we learned yesterday that the AstraZeneca vaccine looks like it has effectiveness in the same ballpark as J&J and Moderna and Pfizer. And then today we learned that there is a group from the NIH that is a bit skeptical about some of the data technologies that were used in that. Now, you've been involved in these studies. How could that happen?
Well, that's a very good question, Tom, and I think is being asked by many scientists and the general public today, and in large part it's a result of data released by press release, and I think that is an inherent conflict when companies are strategizing their communications for press releases prior to the full peer review and publication of the data. And so we see some discrepancies by that approach. And indeed, I think AZ is accurate in communicating that they had a predefined analysis to look at efficacy with a cutoff date of somewhere around maybe Valentine's Day, earlier in February. And as the pandemic has evolved, of course, there are more variants circulating, and the case count has increased since that time. And the DSMB, the Data Safety Monitoring Board, has privy access to data over and above that cutoff date. And AstraZeneca will be, I think, pressured and forced to reveal all of the data when they do pursue the EUA with the FDA. And so that's
the emergency use authorization that would allow the FDA to allow that vaccine to be used in the U.S.
Yeah. And they did state in the announcement that they intend on pursuing this emergency use authorization in the coming weeks. So that's very encouraging news because it will really reveal a much more full deck of the data when they do that.
So right now we've got 3 vaccines in the U.S. that have emergency use authorizations. We've got the 2 RNA vaccines, Moderna and Pfizer BioNTech. And then we also have the J&J vaccine. So Jim, first question for you, because I know this is going to come in. Differences between them. If you had a relative who asked you which one should you take, I know how Larry answers it. I want to see how you answer it.
Yeah, I haven't heard him recently answer that question, so I'll make my stab at it, which is we've been very involved in all three of those vaccine platforms. Well, the two platforms, the nucleic acid mRNA, there are two developers working in that space. There are two developers that we're working closely with in the viral vector space. That includes Janssen and AstraZeneca. And then there's the recombinant proteins, which are upcoming with Novavax and Sanofi. And you mentioned the three who have been granted EUA. I would recommend to any of my family, friends, colleagues, associates to take the first one that they can get of those three that have been granted EUA authorization. And so I've received the Pfizer vaccine, and it was very, very excited to receive that vaccine. I would have gladly have taken the Moderna vaccine, and equally so would have been very happy to receive the Janssen vaccine.
Yeah, and so I had a family member ask me this morning that same question. I answered it exactly the same way that you did. So I'm glad that I got the answer right with that. Can you comment a little bit about the variants? Because, you know, we see the emergence of variants around the world now. The thing I find interesting, Jim, is that the virus has been with us that we know of for about 16 to 18 months, based on where it goes back. And one has to assume that there's been mutation all along, but it seems like the variant issue became really obvious and clear just in the last 4 or 5 months. So why didn't we see variants emerging in April, May of last year? Is it just that there was less virus around to mutate, or is it something about how viruses evolve over time? What do we know about that?
Yeah, I think it's a fascinating question for the evolutionary biologists who are looking at this virus. And I think it is multifaceted. I think that, of course, we're seeing so much more virus circulating now than a year ago. And that's certainly one thing contributing to the increase in variants. The probability of these mutations is that much greater with that much more virus circulating. But we've also, of course, encountered situations where the virus is infecting people who are perhaps immunosuppressed or have prolonged infections despite immunotherapy or vaccination. And this chronic, more long-term infection with the immune pressure driving the virus into mutations that improve its fitness under that immune pressure is something that we're likely to see more of in the future, which is one of the main reasons why it's so important for people to get these vaccines as soon as they can. Because that does, if they are infected, likely shorten the period in which that virus is replicating.
And so, Jim, it's March Madness, and I have to say I know I'm rooting very hard for Gonzaga, obviously, being the team from Washington State. But in the women's bracket, I'm a huge fan of UConn, okay, and their new player Paige Beckers. Now, the coach of UConn is a guy named Gino Auriemma, and Gino got both of his vaccines, okay. And I do not remember whether it was Pfizer or Moderna, but he had 2 vaccine shots. And then about 4 days after his second shot, he tested positive for COVID in the pre-screening that was done of the basketball players and the coaches before they started March Madness. Gino's explanation to everybody was, hey, this is a great example that the vaccines work. I feel fantastic, and I'm hoping to be back coaching as soon as I clear the virus. Others have said, well, maybe the vaccines don't work if Gino still got the virus. What's the story there? How can you test positive after getting 2 doses of an effective vaccine?
Well, I think his case illustrates very well this phenomenon of vaccines reducing severity of disease. And the efficacy of those 3 vaccines that are out there and available for people against severe disease is extraordinary. It's fantastic. Over 90% for the nucleic acid mRNA vaccines and high 80% for the Janssen vaccine. That's just, the best news ever. What we don't know is, nonetheless, this virus occupies predominantly the sinus mucosal surfaces, and the immune system, is not always working effectively in that space. And it may take for full control of the virus for that virus to creep into other spaces in the body, which of course then suppresses the virus and prevents severe disease. But one of the big remaining outstanding questions is really, to what extent can people become infected and also transmit the virus while asymptomatic, while after having received vaccines? And this is very much a continued focus of the CoVPN, the COVID-19 Prevention Network based at the Hutch. And we have an upcoming study in over 12,000 college students that will be focused explicitly on this very question. So stay tuned for a very definitive answer.
And Jim, I want to just emphasize one more point. And please correct me if I'm wrong, but there's nothing in this data that came out today on AZ that should question the safety or effectiveness of the 3 vaccines that are out there, correct? And I think people need to hear that.
Absolutely, that is fully correct. The ongoing safety monitoring of both the nucleic acid platform and Janssen's At26 platform confirm the safety that was demonstrated in the phase 3 efficacy trials, and there were no concerns raised similar to these concerns that are being raised with AstraZeneca. Nothing like that in these other platforms.
Okay, so Jim, a couple of questions have come in I'd like to run by you. Is the opening of schools a good idea?
I think it will be a good idea. It depends on in what location. I do think Texas may have been a bit precipitous in their decision to do so. I have 2 12-year-old twins. We're really excited. They're very excited to get back to school when the teachers and the school districts are ready. And I think if the school district and the teachers are prepared and ready, I think it's reasonable to do so while maintaining certain precautions. And then I'd say, given the fantastic rates of vaccination that we're seeing across the country, with over 2.5 million doses administered every day, we're going to soon get to herd immunity thanks to these vaccines.
And Jim, you're a physician in addition to being a scientist as well. And a question that's come in is, how much protection can immune compromised patients get from the vaccine? And many of our patients at the Hutch have had bone marrow transplants or cellular-based therapies, or they're getting treatment for lymphoma. What can we read into that?
Yeah, I think that's an essential question as well, and one that we'll be collecting and answering in the studies that are ongoing predominantly in South Africa. There are so few participants in these trials who are designated and defined as immunocompromised, and in many of the studies that was an exclusion criteria. So we can't answer the question definitively at this time, but we can say that among HIV-infected individuals, for example, there does appear to be a reduction in vaccine efficacy for mild and moderate disease, but not for severe disease, at least so far from what we can see.
Great. And one last question, Jim, for you before we bring everyone back for the Q&A, which is, any thoughts about taking the J&J vaccine after 1 dose of Pfizer or Moderna?
That's another great question. I'm a big fan of heterologous studies of vaccines. I mean, what's really unique with this platform, 5 of the 6 vaccines that we've been contributing to have used this stabilized trimer that Melody was talking about, and we have an opportunity to look at how one platform as a prime and another as a boost will result in improved, perhaps, immunogenicity, immune responses. And we have another study exactly focused on that question. We don't have the answers yet, but we hope to within a few months.
Well, let's bring Melody and Gavin and Rob back for the Q&A. And we'll start with a question for Rob. And Rob, this question comes in and says that they understand that Dr. Stan Riddell is doing work on how to apply mRNA to cancer. As an RNA biologist, what do you think of that?
It's a great question. So I'm not familiar with the specific work that Stan is doing, but broadly speaking, studying RNA cancer in cancer has already been incredibly fruitful. So in one example, my lab has been working on developing ways to create new therapies for people with genetically defined subtypes of cancer, where these cancers, whether they're leukemias or they're breast cancers or pancreatic cancers, are unified by all having mutations affecting specific RNA splicing factors. And so one exciting possibility is that by developing a targeted therapy that works at the RNA level, we can apply this to many different cancer types that seem totally unrelated. I mean, more broadly, I think it's interesting to think about the huge RNA vaccine developments that have happened in COVID times. And a lot of that technology actually came from the cancer biology space, where many people have been working towards developing cancer vaccines, which potentially could even be personalized to an individual's cancer. So I think there's huge potential there. And I think with the technological advancements that have happened in the last year, we can expect to see continued advances very rapidly.
So Melody, I guess one of the other questions is, can you give us an example of how CryoEM can be applied not just to cancer, because we talked about cancer a little bit, but what about genetic diseases and non-cancerous diseases? How can CryoEM help there?
Well, as I mentioned before, knowing the structure of a protein, how it looks, can be really important for targeting it. The protein structure not only tells you the shape of the protein, but it can tell you some things about the surface qualities of the protein, such as if it's positively or negatively charged, and if it's more likely to interact with things like lipids, like oils, or things like water, other solvents. And so I think one really good example of this is CFTR, the protein that causes cystic fibrosis. So there's a single point mutation in that protein that causes cystic fibrosis. And, even though we knew what amino acid was changed, there was no structure of CFTR, and it couldn't be solved using other structural biology methods. So CryoEM was used in order to determine that structure of CFTR. So now we can also make the mutant version of CFTR, and we can see now exactly where that single point mutation is. And the idea is, since we know where that mutation is on the protein, the context of the rest of the protein around it, the hope is that someday small molecule therapeutics could be developed in order to kind of compensate for that problem with the protein, so it could be fixed in that way.
That's fantastic. Thank you. So, Jim, question came in for you. Two questions, really. The first, and they're related to each other. The first is there any correlation between the level of side effects and the efficacy of a vaccine in an individual? Okay, I was talking to my sister, told me, she said, Tommy, she said, I had an incredibly bad reaction to my vaccine. And I said, oh, I am delighted to hear that. And I don't know if I should have said that, but that's sort of the way I felt. And then another person has asked, related to that, is there any relationship between how badly you might react to a vaccine and whether it implied that you might have had severe issues if you got COVID? Two questions around vaccine reactions and what they might imply.
Yeah, these are both fantastic questions, Tom. And with regard to your relationship with your sister, I'm not going to comment on that. But I'm not necessarily thrilled that she had a bad AEs to the vaccine. It has been a hypothesis, and we've investigated this within the HIV vaccine program, that local reactogenicity would likely correlate with antibody measurement, for example, antibody titers. And oftentimes that does not bear out to be true, surprisingly to us. We thought, the more a vaccine is adjuvanted, the more you see local reactogenicity, and that doesn't necessarily correlate with immunogenicity. And perhaps it does with some vaccines, but in our investigations, that has not borne out to be true. And it does not appear to be true with the mRNA vaccines. And we just looked at it in a preliminary fashion. And only when we have our reliable correlates of protection will we investigate that further. There are some remarkable anecdotes, and I think even on NPR recently, about long COVID patients. That is, people who have had chronic symptoms following their COVID infection. being resolved after vaccination. And that's I think a remarkable hypothesis that it may that vaccination may improve these long COVID conditions, almost in a immunotherapeutic fashion. That also, although compelling, has not been proven with the rigor of a clinical trial. So both of those questions are very good ones, and we'll have answers to both of these. We do see that people who have very little, no reactogenicity still have good immune responses. So if you didn't have a good flaring reaction, that doesn't mean that your vaccine didn't take.
Great, thanks, Jim. So this is a question I have for Rob and Melody and Gavin, which is that you were all in the, I'll say, the early part of your careers. I'm not going to guess how old you are, but you're in the early part of your careers, and we know that everyone from Einstein to Beethoven did incredible work early in their careers. And so the question is, you're all at the very cutting edge of what's happening in genomics and bioinformatics and protein science. What attracted you to the Hutch? What made you want to come here to the Hutch? And we'll go Melody first, Gavin second, Rob third.
There were a lot of factors that made me want to come to the Hutch. One of them was just the excitement that there was for CryoEM here. I knew that I would be in a community where a lot of people were interested in it and were supportive of it, which just makes it a much better place to do research when you're happy and when you have other ideas as well and you can collaborate with a lot of people. Also, the computational resources here are really, really great, and that was really important for CryoEM as well, because I focused at the beginning about the early steps in data collection, but there's a lot of processing that has to go on. So there's a really nice cluster here that will enable us to do this work quickly and effectively.
Great. And Gavin? I think you're mute now. Keep trying.
Can you hear me now?
Now, perfect. Excellent.
Okay, I called in with my phone.
Great.
Because I do want to answer this question, and I think it's just a very important one. And one of the things that really excited me about Fred Hutch is having a dedicated computational biology program and seeing the diversity of the research that's in it, and obviously colleagues like Rob and Melody are in the program as well, and then also the ability to combine experimental work, so we call that wet lab work, with the computational work, at the same time. And so that's something that we're also embarking on and allowing us to really again accelerate our science by doing both ends of it, both the data analysis as well as the data generation, and of course the amazing community and network at UW and SCCA as well.
Great. And Rob?
My answer is kind of similar to the last two you heard, which is I think the Hutch just has a unique scientific community that really recognizes, appreciates, and supports work that is interdisciplinary. So at a lot of institutions, I would be pigeonholed, for example, as an RNA biologist or a computational biologist, and I would only be expected to do a certain kind of study. But what I want to do is work on whatever seems most likely to yield new biology that could actually impact patients. And so we use all kinds of different techniques. And the broad diversity of science at the Hutch really makes that kind of approach possible.
And so, Rob, you're going to get the last question. This is from Casey King. So given the wide-scale availability of compute power and the success of rapid vaccine development, will Bayesian and adaptive clinical trial methods finally reach more mainstream acceptance among clinical trialists?
I think that's an excellent question. I think it's difficult for me to answer, and I think perhaps for a lot of people to answer, because that starts to intersect science with regulation and other sort of broader policy decisions. And so I think it's difficult for me, at least, to say what's going to happen in the future. But I do think it's all been clear to us that RNA vaccines, which have been discussed and touted for a long time, are now here to stay, and they're going to change a lot of things beyond just COVID.
Rob, I want to thank you very much. Melody, Gavin, Jim, our ASL interpreters, you folks have been fantastic. Thank you so much for coming. I also want to thank Laird Norton Wealth Management for your support to enable these programs. Absolutely spectacular. I also want to thank all of you who've donated to our work. Your support is what allows us to do this. As all of you know, the grants that we get from the federal government support about 70 to 75% of our work. We really do rely upon the support from people who help enable the research at the Fred Hutch. And I want to let you know about a special opportunity. Until March 31st, all gifts to the Fred Hutch will be matched thanks to a $250,000 matching gift from the Krabby Beach Foundation. If you're inspired to support the research happening at the Hutch, whether it's cancer or it's COVID, now's a terrific time. Our next Science Says will be April 27th at 9 a.m. Pacific time. We'll be talking about how we're using the power of the immune system to create personalized cancer therapies. Again, one of the 6 important pillars of the Fred Hutch. I look forward to seeing all of you in April. Thank you so much and have a fantastic week.